evening, good afternoon, good morning, depending on where you are in the world. Welcome to the 6th webinar of the series Biodiversity Conversations, India's Opportunities and Challenges. These webinars focus on various aspects of the National Mission on Biodiversity and Human Well-Being. This webinar series is jointly organized by the Office of the Principal Scientific Advisor to the Government of India, the National Biodiversity Authority, and the Biodiversity Collaborative. My name is Ravi Chellam. I'm CEO of the MetaString Foundation and a member of the Biodiversity Collaborative. Today, I'm here to assist Dr. Kamal Bawa, the moderator of this webinar. Before I introduce him, I have a few announcements to make. Please open your chat box as text messages would be sent there so that you can follow what are the announcements. We've already received some questions and comments by email and welcome additional questions and comments during the course of this webinar. Everyone is requested to use the question answer facility to ask questions to the panelists. Questions that are not answered during the webinar due to a shortage of time will be answered via email, and you can also get in touch with us by email at contact@biodiversitycollaborative.org. Questions can even be asked anonymously if that's your preference, but we encourage all of you to include your email ID while sending your questions to enable us to respond to you later, just in case if your question hasn't been dealt with during the webinar. This webinar is also being live streamed on Biodiversity Collaborative's Facebook page, and a recording will be uploaded soon on our YouTube channel, which is again Biodiversity Collaborative. We gratefully acknowledge the support we've received from the Office of the Principal Scientific Advisor to the Government of India for conducting these webinars. During this webinar, the chat facility will be disabled. Please do not use the raise your hand function. We request all the attendees only to use the question answer function to send your questions. It now gives me great pleasure to introduce Dr. Kamal Bawa, President of ATRI and a member of the Biodiversity Collaborative. He's a Fellow of the Royal Society, Fellow of the American Academy of Arts and Sciences, and Fellow of the American Philosophical Society. He's a Distinguished Professor of Biology at the University of Massachusetts, Boston, and a recipient of the Linnaean Medal. Kamal, over to you.
Thank you, Ravi. Good afternoon, everybody, and welcome to this webinar on exploration and discovery and genetic characterization of biodiversity. I'm honored to be a part of this very distinguished panel with Dr. Mao, Director of Botanical Survey of India, Dr. Karash Chandra, Director of Zoological Survey of India, Professor Umar Ramakrishnan, and Dr. Catherine Workman from National Geographic Society. I will introduce each of them a little bit later, but I just wanted to mention that it's really a privilege to be participating in this webinar along with these people. Somebody has already commented that they are very glad to see the Director of Botanical Survey of India and Director of Zoological Survey of India in the same webinar. So we already have received very good comments, and we have actually managed to bring in National Geographic Society too. You know, the world's premier organization in terms of exploration and discovery. And we are very grateful to Catherine Workman for making the time to be here. So without further ado, I would proceed with my very, very brief presentation. And I think, as I mentioned in the first webinar, the biodiversity mission has 7 or I should say 8 components. And I think you have been hearing about various components, and I'm going to focus today on this component, NISARG Bharat. And this component basically consists of 3 subcomponents, one dealing with exploration, discovery, and genetic characterization of India's biodiversity, and that will be the focus today. National Framework for Electronic People's Biodiversity Registers, and we have a forthcoming webinar on that aspect. And Cataloging and Mapping Life of India, we will have another webinar on that. Basically, NISARG Bharat is devoted to compilation of databases, mapping, having very comprehensive and integrated databases. Not only the biodiversity databases, but also databases, and more importantly, information systems associated with other components of the mission. I want to make a few comments on India's biodiversity. We all know that we have 4 global hotspots of biodiversity. We are one of the 17 megadiverse countries of the world. And although we constitute 2% of the world's land area, well, we have approximately 8% of global biodiversity. So if we extrapolate the figures that are available for the number of species in the world, we could have probably 800,000 to 1 million species of plants, animals, and other organisms. We should keep in mind that only about 190,000 species or so are named. This is an upper limit. The number could be just 150,000 or even less. When you go to the literature, it is very difficult to find how many species from India have been named. But I think roughly, maybe somewhere between 150 and 190,000. So that means that a vast majority of the species, probably 300, 400, 500,000 from India have not even been named. And I want to focus particularly on plants for the next couple of minutes. Because in India, when we think of biodiversity, we think of our very charismatic mammals, tigers, lions, elephants, and of course they are beautiful. They're lovely and cuddly, and who wouldn't think about those when you think about biodiversity? But I want to stress, plants are the basic structural elements of the ecosystems. Plants define the basic features of ecosystems and communities. Plants provide the habitats for all life forms to exist, and they constitute the base from where trophic interactions emanate. So we have to pay a lot of attention to plants, our ecosystems, plant committees, and so on and so forth. Now, in terms of plants, I think there are probably 28,000 species of plants, and people will argue that that is not true. Your guess is as good as mine. Dr. Mao might have different figures. He's more experienced, so I will concede to his estimates. And this 28,000 includes probably 10,000 or so algae. There are approximately 1,200 species of ferns, so there might be 1,300. a few hundred species of bryophytes. Probably the flowering plants may be around 18,000. There are some estimates 16,000. And again, Dr. Mao will talk more about it. I want to mention something about fungi. We have only about 10,000 or so species of fungi described, but there are possibly 100,000 species of fungi. And of course, Northeast India is the center of this biodiversity. Approximately, I would say, 1/2 to 2/3 of India's biodiversity is in the Eastern Himalaya, a very, very important region, Northeast India. Probably a couple of hundred species of impatiens there. Orchids, the recorded number of species is perhaps 900, 1,000, but I think the number might be eventually 1,800, 1,900. Again, Dr. Mao is an authority on orchids. He will have better estimates. Primulas, number of described species is perhaps 100, but maybe there are 200 species. Of course, rhododendrons. One of Dr. Mao's favorite genus, and mine too, and Uma's too, and Catherine's too, everybody's. I want to say a few words about rhododendrons. I think it's not only the species, when we want to do biodiversity assessments, it's not only the species we want to name and describe, but the various interactions. And Uma will talk more about it. We have very little information about the distribution, abundance of these species. Species have been recorded maybe 80, 90 years ago. Do we still have those species? So a lot of work needs to be done. And take, for example, rhododendrons. These are 4 different species at 4 different altitudes, dominant features of 4 different types of communities. And that, I think, suffices to prove my point that we have to pay a lot of attention to plants and vegetation when we are talking about biodiversity of India. So let me quickly mention what are the critical elements of the exploration, discovery, and genetic characterization of India's biodiversity subcomponent. My fellow speakers will talk more about it. Undertake field explorations, deploy and develop molecular tools such as DNA sequences and environmental DNA to develop digital databases in collaboration with EPBR, CML, and ongoing programs of BSI and ZSI and other organizations, and engage citizens in biodiversity assessments, and strengthen BSI, ZSI, and other centers of taxonomic research. And I have a few questions for the panelists. I would like them to address. What are the major gaps in biodiversity assessments? What are the challenges in filling these gaps? What are the conceptual and technological innovations that can help in rapid assessments of our very precious and declining biodiversity? And how can we engage thousands, if not millions, of students and citizens in biodiversity assessments? And with those comments, let me introduce Dr. Mao and Dr. Karash Chandra. Dr. Mao is the director of Botanical Survey of India, a 130-year-old institution, which is country's premier Center of Plant Taxonomy, Survey and Conservation. We are delighted to have you, Dr. Mao. And he is the first person from the biodiversity-rich Northeast India to head the Botanical Survey of India. And he has worked on orchids, rhododendrons, medicinal plants, ethnobotany, and conservation of rare, endangered, and threatened plant species. Dr. Mao, welcome.
Thank you very much, Dr. Kamal Bauer. And also thank you, Dr. Ravi Chalam, for giving me this opportunity to share the role of BSI in this national mission for biodiversity, which is a very important and which is what BSI can play a role. And in today's seminar, this webinar on exploration, discovery, and genetic characterization of India's biodiversity, I just like to highlight the role of BSI, what we can do for this mission. So that's why I will just go briefly because the time is short. So just I have prepared only 26 slides, and I hope it will be sufficient to just highlight the activities of my department. So let me start. From the beginning, no? No. I think it's to start from the beginning. Okay, this already Dr. Kamal has already told about the establishment of BSI, but our mandate, I just wanted to give you the mandate. Our mandate is to undertake intensive floristic survey and collect accurate detailed information on the occurrence, distribution, ecology, and economic utility of the country. Then to collect, identify, and distribute material which may be of use to educational and research institutions, and to act as a custodian of authentic collection in well-planned herbaria, and to document the plant resources in the form of local, district, and state national flora. This is the mandate of the BSI. So for this, the government has set up regional centers. And today we have 11 regional centers in all parts of this India in different geographical zones to undertake the mandate of the department. So here we have, like for the Himalaya, Sikkim is there. For the Northeast, Shillong, that is Meghalaya in Shillong. Then for Arunachal Pradesh, we have a center in Itanagar. Then we have in West Bengal, basically, like what I would like to say is that like for Shillong Center, Eastern Regional Center, we are looking at that center is looking after 6 states, all the northeastern states, apart from Arunachal and Sikkim. So it covers 6 states. Then for like in Calcutta, we have this headquarters is there, and 4 units are there, like Central National Museum, Indian Botanic Garden, Indian this museum is there, and then CBL is that Central Botanical Laboratory. And then this center here from Calcutta, from CNH, it look after like Bihar, Jharkhand, and West Bengal. So 3 states are covered. Then we have in Dehradun for Northern Regional Center, this cover of 2-3 states again. And then we have recently set up one in Solan, that is Himachal Pradesh, for the high altitude regional center. And that is going to look after Himachal Pradesh, Ladakh, and Kashmir, these regions. And then we have for western side, this Jodhpur is for Arid Zone, the regional center. That looks after Rajasthan and Gujarat. And then we have in Western Regional Center that is in Pune, and that looks after Maharashtra, Karnataka, Goa, these are the regions it looks after. Then we have set up another center in Hyderabad, and that is looking after this Telangana, then Andhra Pradesh, Orissa. So 3 states are looked after by that. So then in Coimbatore, we have the Southern Regional Center, which look after Tamil Nadu, Kerala, and Kerala like that. And we have in Andaman and Nicobar Island in Port Blair for Andaman and Nicobar Island. So like that, we have set up all over India in different geographical zones for study of this to achieve our mandate. So now, so far, since the reorganization of BSI in 1954, BSI scientists have discovered 1 new family, 42 new genera, 1,719 new species, subspecies, and varieties, and inventorized around 1,900 rare, endangered, or RET species. And BSI have already documented flora of this 7 biosphere reserve, 95 national park, 268 wildlife sanctuaries, 27 tiger reserve, 28 state flora, then 26 districts. These are published. And from this map, you can see the well-explored, the percentage of exploration in different parts of India. So the first one you see the light green, yellow, that is well explored, or more than 70%. Then the pinkish one explored 40 to 70%. Then the last one is being explored, or this less than 40%. So that's why in many parts of India, still many places are to be explored properly. Even though BSI has been there for the last so many years, we have not been able to explore properly yet, thoroughly. So here, if we see the comparative account of plant diversity in world and India, as per the latest figure of BSI of my department. So we can see here, this is the figure, what we are saying, the world and India. In a vascular plant, so far we have 20,189. And flowering plant, we have 18,882. Then algae, 7,434. Bryophyte, 2,786. Pteridophyte, 1,307. And gymnosperm is 82. So it is coming to 50,680, the total of this plant diversity in our country. So if we look at what BSI have in our BSI harbouring holding and status of this digitized in different regional centers or units of Botanical Survey of India. If you see here the, I don't know whether this will be, this Andaman, the first is Andaman in Port Blair, that we have about 42,000 and type 404 is there. Then this data in MS Excel data is already that much, 23,000 is there. So the images also, which is digitized, is about 28,000 is that. The next comes is the Arunachal Pradesh, that APRC. Here, 33,388 the herbarium holding is there, and the rest, the figure is that is there, you can see. Then SZRC is the Jodhpur for Arid Zone Regional Center, that is in Jodhpur. We have there about 37,000 herbarium specimens are there, and the other figures you can see the data. Then BGR, that is of course, we have set up a garden in Noida in Delhi, near Delhi. Here also they have started collecting herbarium plant specimens. So they have that is a very small collection, 2,000 only. Then the CBL is here in Calcutta, that is Central Botanical Laboratory. They work mostly on economic plants. So they have a collection of these economic plants. There's about 12,000 herbarium specimen, and they are the then CNH, the Central National Herbarium, that is the largest herbarium in our country, and not only in our country, in Southeast Asia. So that we have 25 lakh plant specimens collected and kept there. So at here also type specimen itself is 22,000. And then data which we have make into Excel sheet is 71,000. Then images is 71,189 is there already. Then Central Regional Center, that is in Allahabad. And this also harboring holds about 150,000 specimen. Then the next one is the Deccan Regional Center. This is a very new regional center in the Hyderabad, and that was we have so far now about 30,000 harboring specimen is there. Then the next one comes Eastern Regional Centers in Shillong, and that center holds 147,113 specimens are there. And you can see the other data is there. The Indian section for the Industrial Museum, that is in Calcutta, this center also have a very good collection of economic plants and industrial plant uses of plants. About 70,000 specimens are holding here. Then Northern Regional Center, that is in Dehradun, which was looking after all this Western Himalayas and Central Himalaya. Now they have a collection of 125,000 harboring specimen. Then Sikkim, that is Sikkim Himalayan Regional Center, that has about 57,000 collection. Then Southern Regional Center, that is Coimbatore, it has about 2 lakh specimens are collected and kept there. The Western Regional Center, that is in Pune, we have about 1,79,000 specimens in the herbarium. And total, what BSI has collection of this specimen is all over India in our collection, 35,85,986 specimens are there. So that is, you can see the thing. And the digitization or that data in MS Excel sheet is 3,14,210 is already there. Then for images, whatever images, photo images of herbarium or this thing, 1,68,492 so far we have already done. So here in the publication, our main focus was on the flora of India. So this thing, the flora of India was planned for 32 volumes. And this started somehow quite many years back. I had about 10 volumes have been already published. And then the remaining 22 volumes were completed by this 21-22. The plan is that we have brought out the checklist of flora of India in 3 volumes. Volume 1, 2 is angiosperm, and volume 3 is monocot. Dicots, volume 1, 2 is dicots, and volume 3 is monocots. So it is coming to 20 plus taxa. Then this flora of India, we have taken up in an action mode to complete it by 21-22. So this year, by December, 11 volumes will be ready. And then by next year, another 11 volumes will be ready. So for this, about 80 people are participating in the job to complete this thing. Because earlier, all the manuscripts were, many people have, this has started long time back. So manuscripts are there. So now we're updating and we're trying to finalize. So that is why we'll be able to do by next year at the 21-22. So here, so far, we have published 29 volumes of fascicles. Then state flora, as you see here, for 49 volumes and of 28 state published. And this in the red 41 volumes and of 24 states to be published. These are almost ready, but not yet published. So then district flora, earlier we were doing district flora, but this has been stopped. Now we're not doing any more district flora. Then this other miscellaneous publication like red data books, this also we are supposed to do more. We have done only 3 volumes, and in midway it was stopped. Then again, these other publications or this other thing, you can see a glance of this is our publication, what we have been bringing out. So here, again, when it comes to plant taxa discovered as new to science in the past 10 years, if you look at this chart, or if you this bar diagram, you see from 2010 to 19, you can see this is how it has increased. And then you see this in 2018, 19, this has jumped up like this because what happened was that this many new species discovered from fungi. And in fungi, so to say, in 1 year, more than 100 or like that. So again, the trend is that most of the discoveries are coming from fungi, lower group. That is what you can see here. So this shows that lot of work is still remain, and the more we do research, the more new species are coming. And these are, of course, some of the GSI's activities, which is you can see online journal we have, eFlora. eFlora, we have put up 8 volumes which has been completed, and the rest, whatever we complete, will be put up. So in the next 2 years or 3 years, we'll have all the flora of India, all the 32 volumes in online, like what the Chinese have. So that will be there soon. Then we have website updated and all the thing. Then all the other materials, we are working with the NIC to complete it. So they have told us.
Dr. Mao.
So by within 6 months time, I think we'll be able to complete the updated. So also we're also working on the rate listing. So some of the things, which example I'm just giving here. And then there's the same thing on the especially orchid. Our people have done for this Northeast. Then the CITES also on this non-determined finding of NDF plants. We have work on it. So these things also a lot of work is left to do. And then here, one of our main mandate is also this on ethnobotanical studies. So here we are also working on nutraceutical and toxicity studies of wild edible fruits and vegetables. So our people, our team is working on that. Then of course, extensive conservation is one of our mandate. So we have all the regional centers, botanic gardens, and these are the activities we're also doing. And then, of course, tissue culture is there, part of that for the conservation purpose. Then, of course, we have set up molecular taxonomy lab in 2-3 places. And the work we have also started to assist the taxonomies in confirming the species and also for better publications. Otherwise, many of the international journals are refusing to publish the new species. Also, we thought this DNA.
Dr. Mao, we have to wrap it up, please. Thank you.
Then these are the other publications of algae, then this fungi, then this is lichens and bryophyte. I must say that these lower plants were short of manpower. So in this area, a lot of work is to be done yet. Because right now, we have in each section, in each group, pteridophyte, only 5 experts are there. Then algae, 5. Then lichens, at present, only 2 experts are there in the department. So that's why we have a really shortage of staff is there. We also have been to Antarctica 10 times. Our scientists has gone for study, so we also have work on that. Of course, these are outreach activities. I think I need not say all these things. So the challenge is for us is to strengthening of manpower, then infrastructure, and expansion of regional centers, because we have so many regional centers, 11 regional centers. Each center is looking after 3-4 states in some regional center, but we're having 7-8 scientists. So to look after 3-4 states with 5, 6, 7 scientists is very difficult, and that's why we cannot do justice to that. So this is what I just wanted to thank you so much for giving me the time.
Thank you, Dr. Mao. We move on to Dr. Karash Chandra, who is the director of the Zoological Survey of India, a 100-year-old institution. And Dr. Karash Chandra has specialized on insects and is a leader in terms of the surveys and conservation of insects. So Dr. Karash Chandra, if you could keep your comments to the agreed upon time of 12 minutes so that we have some time for the other speakers. Thank you.
You're muted, Dr. Chandra.
Yes, muted. No.
So, Dr. Kamal Baba, Dr. A. Mao, our Director, Botanical Survey of India, Dr. Uma Ramakrishnan, our Associate Professor from NCBS, TFIR, Biodiversity Collaborative, Dr. Catherine Walkman, Senior Director and Scientific Advisor from National Geographic Society, Dr. Ravi Chalam, who is coordinating all this program. Thanks for giving the opportunity to discuss about Zoological Survey of India, what the role we have for the National Mission on Biodiversity and Human Well-Being. Dr. Kamal Baba has already discussed these three sub-programs which are to be undertaken by Botanical Survey of India as well as Zoological Survey of India. This is exploration, discovery, and genetic characterization of the India's biodiversity, national framework for the electronic PVR, cataloging and mapping life. This has already been elaborated, and I will not go into the detail about this is again the key mission objectives which have already been included in DPR. This is the details of the last three components which we are going to take up. But out of these three components, now the task for the present webinar is to cover the first topic, and that is what now we are discussing among the stakeholders, BSI, ZSI, and then NCBS, who are the collaborator. I will just go into a little brief information what the faunal diversity we have in our country. Altogether, by 2019 December, the database which we have updated from Zoological Survey of India, this gives the information of 102,161 animal species pertaining to 3,076 families. This number we are just sharing for the first time. Nowhere it has been published that these number of the families are known from the India as far as all 26 phylum of the animal recorded from the country. This includes 76,461 species of the arthropods, and within these 102,000 plus species, there are 28,527 endemic species. So they are the sovereign rights for the country under Biodiversity Act as well as Biodiversity Rule 2004. And the results of these, all our species which are included, have been published by Zoological Survey of India in 1,550 documents and around 12,500 articles. They have been published in national, international journals as well as the Scopus journal. So as far as this documentation and the area and the different state protected areas, then biogeographic zones, that I just want to brief here before I go into the detail about the three components. Out of 36 states and union territories, 25 states have already been surveyed and documented. That detail I'll just share with the next slide. And India's landmass has been classified into 10 biogeographic zones as per the classification by WII, Roger, Pawar, and then Dr. Mathur. They have given the classification. And the faunal resources of the country of these 10 biogeographic zones we have been doing for the 1 decade. And the Himalaya is having now 30,377 species. Trans-Himalaya, which is also known as the core desert area, 3,324 species. Islands, 11,009 species. The Northeast, to which our Dr. Bao was mentioning that 2/3 are about more than 50% of the species of the flora are known from the Northeast. But as per our entire complete list of the fauna from protozoa to mammalia, which we have documented, and it's very soon, I think, going to be published in one of the document, 18,527 species have been documented only from the Northeast. We have the details about all the states of the Northeast states. Desert, which is there in the western region, Rajasthan, some part of the Gujarat, 3,324 species. Then semi-desert, starting from the Gujarat up to the Madhya Pradesh, Rajasthan, Punjab, Haryana, covering 7,424 species from coastal region. Starting from the West Bengal to the Gujarat, it's having 11,803 species. So this is just about biogeographic zone. There are 2-3 biogeographic zones which are still left, that is Western Ghat, but we are estimating around 14,000 species are to be known from the Western. So after having this information, I could say it very clearly that Northeast is having the second most diversity if you compare the whole 4 biodiversity hotspots, and the Himalaya is having the highest the faunal diversity in India, followed by the Northeast. Then 3rd is the Western Ghat, and the 4th is Andaman Nicobar Islands. Then there are several ecosystems like marine, freshwater, then estuarine, that has also been documented by JDSI. We have 20,444 species from the marine ecosystem, 9,457 species from the freshwater, 3,939 species from the estuarine, and 5,747 from the mangrove ecosystems. These documents have already been published by JDSI. There are 903 protected areas which are covering around 5% of the total geographical area of the country. We have already documented around 120 protected areas. These protected areas include national park, wildlife sanctuaries, conservation reserve, as well as the community reserves. But there are many other conservation areas like tiger reserve, biosphere reserve, Ramsar sites, that so on. I will just discuss in the later stage. For the effective conservation of the water bodies, 37 wetlands under the category of the Ramsar sites have already been notified. 10 Ramsar sites have been added just last year. And from there also you could see that 6,622 species have been documented, and they are available with us, the complete list. Apart from that, there are several 18 biosphere reserves are there, and those also have been documented, and at least 10 biosphere reserves have been documented. As far as the faunal explorations are covered, what are the gap areas? We want to discuss at the today's meeting. As far as explorations are concerned, from 1916 onwards, ZSI has been exploring throughout India, the states, protected areas, ecosystems, biogeographic zones. So for last more than 104 years, I could say, there had been 3,500 extensive surveys, and now we have 5.6 million specimens comprising to all the group of animals, starting from the protozoa to mammalia, of which 3.8 million specimens have already been identified. So if you see the area wise, what are the gap area? If you see this, our Survey of India topo sheet, which is 1 into 150,000 grid. So there are around 5,000 plus our grid in this size. So that at least around 45% have been covered, and 55% of these grid of the size are still, we do not have the record of a single to any number of the species. And so that we can say, still, although there had been a lot of extensive surveys, but as far as intensive collection and surveys are concerned, hardly 20% of the area has been covered, as far as the whole country is concerned. How we are going to take up these surveys now, if we just be a part of the this mission, we are going to take up the survey through our different methodologies, because unlike the plants, just have the collection for different flora and just preserve it. We have more than 26 phylum, and each and every group of even insect has got different methodology for the collections. So we'll be having application of the modern tools and the GPS, geomapping, artificial intelligence, mobile app. Already we have started, or people have been trained to have the mobile app in the field, and they would be getting, it's not 1 or 2 parameters, around 30 parameters are there, which will be collected by the, and then we will be doing the modeling with the similar habitats, all those species are present in other habitat or not. Then molecular genetics, for last 1 decade, our scientific team has been working on the molecular genetics. So these are the modern tools which will be just taking up. Fauna of India, already 141 fauna of British India and fauna of India after 1947 have been published, and that comprises itself 45,000 species. So before independence and after independence, this 141 fauna volumes includes these many species. As far as this fauna of India, whatever the remaining species are there, we have to just survey and document. We may be requiring more than 200 experts and for the 200 groups, and then at least some of those groups could be covered. State fauna, already 25 we have completed. Biogeographic zone, I have already discussed it. Out of 718 districts which are already present in the whole country, we have a database of 100 species may be varying from 30 to 1400 species. And the conservation area, 903 protected areas are there. We have information about. This is actually after having the National Zoological Collection of entire ZSI, comprising 5.6 million specimens, and having the entries of the around 4.5 lakh units. They have been all geotagged, and you can see that out of 5,160 our grids, we have the data on the 3,537. This is again the entire, if you just see the details, so the number of species in each grid, how many are there. So this is the relative actually number of species ranging from 1 to 100, even 200, even 2,000 species one could find from one of the grid, which is the size of the 27 into 27 kilometer of the 715 kilometers square. This is the number of species which we have already documented and published from the different states, 25 states. Then discovery, how we are going to do it. As it has been mentioned, around 8 million species are expected, 8.7 million species for the plants and animals. But within India, what we are going to do it. If with the same pace we are going to it, so it may take another 800 years to describe and record all these species. But in India, we are expecting around 5 lakh species of animals. And for that, we have to have the strategic plan, how many, but we are expecting within the next 10 years during this project, at least 10,000 to 15,000 species may be described new to the science. If we have around 200 scientists only dedicated for this purpose. These are the new discoveries. Along with the BSI, we have been published. So average number is around 200 species, new species as well, the new records starting from 2019 to 2018. And additionally, we have 317 species per year. That is for the new record as well as if you. This is about the status of biodiversity shortfall. That is what now one of the objectives we wanted to discuss is. So although here I have mentioned all 7 biodiversity shortfalls, but today I think we have to give more emphasis on the Linnaean shortfall, and that is about the species taxonomy. So as per my estimate, and I have taken the viewpoint of my colleagues also, if we understand there are the 5 lakh and plus more species which might be occurring in our country, so we are still lacking the database of, as far as exploration is concerned, from 85 to 90%. But if we understand about the 1 lakh species, then the database about geographic distribution, evolution, abundance, ecological function and traits, abiotic tolerances, ecological interactions, it varies from 90 to 90%, 80 to 95%, where we have the gap areas. And these are the approaches how we can do it. This is again the Linnaean shortfall. India is maybe having around 5 lakh species, and we have been publishing it. So at the most, we can have the 200 species so far in the normal pace, and with the new records, it may be around 300 species. But every year, at least, if we have at least 1,000 species could be described with the support of this particular mission. This is genetic characterization of India's biodiversity. And since the establishment of
Chandra, could we wrap it up in 1 minute, please?
2 minutes, sir. I'm just wrapping it.
I think we will lose the audience.
Yeah, I'm just
finishing by ourselves. Thank you. Thank you anyway.
It's almost the last 1 or 2 slides.
Okay, thank you.
So, since the establishment of molecular systematic laboratory in Kolkata, and generation of DSI has been taking up, already we have 6,000 plus DNA barcodes which have already been uploaded in NCBI database. And now we are expecting around 1 lakh barcodes by 10 years pertaining to 30,000 species with the support of this mission. So if you compare the database of the whole India, we have the database which have been given to me. We have at least 35,381 barcodes available in the database, of which comprising to 8,000 plus species. Of this, 1,155 species is from ZSI1. So pertaining to different group, you can see starting from the insect, then vertebrates, and many groups from protozoa to mammalia are there, and 6,500. So with regard to this genetic characterization, how we are going to proceed it, the main component is that by next year, at least 10,000 per annum, as well as the 3,000 species, we may have digital sequence information. We may also proceed to the conservation genetics of 50 species, then meta systematics for 1,000 samples, genomics, which we are going to take up as a whole genome, mitochondrial genome, then amplicon sequencing, gut metagenomics, that around 50 per annum, and the 500 total, we would be able to achieve it. So this is just and with this, I just thank all for just having the patience. Thank you all the panelists.
Dr. Karash Chandra, thank you very much for a very rich presentation. And clearly, we need another webinar so that we can go into the details presented both by you as well as Dr. Mao. We are very grateful to to both of you for these very rich presentations. And we certainly will come back, and there is a lot to discuss. And let me move on to Dr. Uma Ramakrishnan. I think she is going to help me in managing time, first and foremost. And she's a professor at National Center for Biological Sciences in Bangalore, and she works in the area of conservation genetics, biogeography, and emerging infectious diseases. Welcome, Uma.
Hi, everyone. Thank you. Thanks for having me on this panel. And, I guess my role here is to talk about novel technologies. Exploration, is fascinating to all of us. And it turns out that over the last 20 or so years, explorers have a new technology in hand, one of which I will discuss a little bit. So we're all like any of us, you wake us up in the middle of the night, we know that the distribution of diversity is not equitable across the earth. There are these biodiversity hotspots, not only described for having high biodiversity, but high threat and potentially high human impacts as well. And while this is important to know, it's also very true, and you just heard about this. Many of the shortfalls from Dr. Chandra's talk were in the 90%. That's crazy, right? So we seem to talk a lot about this place is biodiversity rich, that place is not, and so on and so forth. We really don't have a very good knowledge about biodiversity. And this is evidenced by the fact that the rates of discovery of species are high, and you saw that also in Dr. Chandra's talk. Yeah, and this is particularly true in India. We are still discovering many species. Along with this, there's been a lot of changes in taxonomy, new integrative approaches, which include, for example, tools like genetics and genomics. So, I think one of the most fascinating things in recent times have been these techniques, which I'll talk about very briefly. So this is a picture of a lynx, and you can see the lynx is walking on the snow. And it turns out that, if you actually go there and collect some of that snow and analyze it in the lab, you can actually get DNA from the lynx. So if you were to just walk over to this place, collect some snow, you could figure out who has actually been there. So this kind of exploration, exploring the unseen in a way with DNA and genetic sequences, is something which is started only very recently. And I think it's really exciting to be able to just go into a place, pick up stuff, come back to the lab and try to understand biodiversity without necessarily seeing it. Not to say that traditional taxonomy is no longer important, but these are new methods which can be integrated with those traditional approaches. So the bottom line is that DNA is very stable, right? So it stays in the environment for very long, thousands and thousands of years, and trace amounts can be used to amplify it or to copy it, pull it up, pull it out, and read. And because DNA sequences are unique, we can then use whatever we read compared to a database to figure out what it is that we have. And several groups are already using these approaches. Scientists at the ZSI, Wildlife Institute of India, and Lacan CCMB are already, and I'm sure there are others, these are the ones I know of, already working to use environmental DNA approaches to investigate biodiversity in lakes and rivers, potentially even looking at ecosystem processes through proxies of biodiversity change. And while this will give us a lot of information on what might be present in a location, it might give us lists. I think it's really also important to move beyond these lists and think about communities and maybe how are things interacting in these sets of species which we may uncover. So for example, what are patterns of biodiversity? We heard about the biogeographic regions, and this is something really fascinating, but also how does this relate to ecology? So for example, if you walk up a mountain, do you find diversity changing? We expect diversity to increase and then decrease, or just increase or just decrease. How are these patterns across the various mountain ranges in India? And so on. So basically understanding not just the numbers of species or how they are in an ecosystem, but what are patterns of diversity, and how do these patterns of diversity vary across space, and how might they change across time if we understand a bit more about species. There's also, of course, really important approaches here, which I'll get to a little bit later, in terms of change and how species may be affected by changes in land use and so on. So can we basically use these novel technologies, quick technologies? It's very easy in a way, right? To just go quickly pick up some mud or some whatever and come back, water, and quickly sequence everything. So if you can do something like this, can you really take that one step further and really understand more about biology and ecology in those locations? Also, what about the evolutionary processes which have generated these species and their distributions? So I'm just giving you a very a simple example here. It's a toy example. We might expect that species richness, which is basically the numbers of species in a location, would be different, right? Across, say, all the grids which I have put onto this map of India. At the same time, the phylogenetic diversity or the evolutionary history as a whole, kind of consolidated into the species that live in a location, into a community, will also vary. And so what we might expect though is where there are lots of species, there will also be a lot of evolutionary time or a lot of evolutionary history that may be maintained. And that's what we would call a hotspot, where you have high species richness and high evolutionary diversity of phylogenetic diversity. On the other hand, you may have other spots or locations which are cradles, where lots of species are being generated. So there's a lot of recent speciation, but at the same time, it's recent. So the number of species is high, but they are not very evolutionarily distinct. On the other hand, you could have museums where you have a lot of old lineages, but there aren't that many species. So species richness is low and evolutionary diversity is high. And this map just shows, for example, for mammals, endemic mammals, it's just like I said, it's an exploratory analysis, not to be taken very seriously. In white are protected areas, and you can see marked here cradles, hotspots, and museums. So can we then assess with assessments of this diversity and their evolutionary lineage, evolutionary affinities, whether we are actually protecting hotspots, cradles, museums, what's going on? So I'm saying, with evolutionary understanding, we can take this listing of biodiversity several steps further in also trying to basically understand and prioritize conservation to see what we can protect and how the species can be used sustainably. So this is, for example, unprotected museums, or you can have unprotected cradles and so on. So, of course, it sounds like magic, right? To just go pick up some snow and find the links. But there's a lot of issues with such approaches. And of course, the first question we must always ask as scientists is how robust are these inferences? What if I just picked up some DNA, blasted against a database, and it says, whatever, wildcat from Europe? That's probably unlikely, but that might happen because most of the databases are filled with species information from other places, right? So how do we look at what might be issues with such approaches? Can we use artificial intelligence or machine learning approaches here? How do we control for how long a DNA may stay in an environment and so on? So along with understanding these biological processes, there are many methodological and technological in terms of informatics approaches which are also important. So I think this is my 2 cents. The keys to success of such a large activity will for sure involve these novel and quick technologies, but also a lot of analysis and strong quality control to make sure that we are really estimating things correctly. And I think it will be really fun and really important to have citizens participate with us through these programs, and of course have grants for the community across the country, regardless of whether they work in NGOs, government institutions, research organizations, universities, to collaboratively together work on these issues. So I'll stop there, and I'll stop sharing my screen. And thanks, I hope I didn't take too much time.
Thank you, Uma. I want to assure the audience that we will have some time to answer the questions. We hope to answer or respond to at least 4 or 5 questions. So be patient. We may have to extend the time by 5 or 10 minutes, but we will come back to the questions. They're very, very interesting questions, and some of the questions are very common. A lot of people are asking the same questions, so we are going to pick up the questions on the basis of those criteria. Our final speaker is Dr. Catherine Workman, And as a Senior Director of Scientific Grants at the National Geographic Society, Catherine has been overseeing the strategic development and the management of the Society's research, conservation, and technology grants program. She has been with the Society for 8 years. And welcome, Catherine. I look forward to your comments.
Great. Well, I thank you. And I recognize that we are up against the time here. So I'll just make a few comments, and I don't have a presentation to share. First of all, thank you. Good morning from Washington, D.C. Good afternoon in India. It's really my pleasure to be here, and I extend my appreciation to the Biodiversity Collaborative and especially to Dr. Kamal Bawa for the invitation to be a part of this seminar and to join such distinguished panelists. So thank you. I think at this point in the morning or the seminar, most of the salient points regarding the importance of biological inventories to fill the Linnaean shortfall in India have been made. So the relevance of species exploration and discovery goes well beyond scientific curiosity and our fundamental desire to understand the planet and its inhabitants. This is necessary. Biological inventories are necessary for improved natural resource management, for identifying species with potential benefits to humanity. And the importance of such efforts have really never been greater, given the accelerated loss of species of biodiversity that the planet is experiencing. We are losing species before we realize the potential ecological and economic relevance of undiscovered species to humanity. So it's a timely endeavor. And certainly I agree with the panelists on this front. I'd like to offer briefly two other reasons why I think focusing on species discovery in particular is an incredibly valuable approach. And the first reason here is that it's positive. Species discoveries are a fantastic way to excite the general public about the natural world. It's a tool for curiosity, for education, for exciting people to the intrinsic value of non-human life. Species discovery fuels conservation optimism, and it's a hook to engage early career conservationists, students, funders, as well as the general public, because of the fact that it's fun and exciting. We all know in order for us to protect the planet, to conserve planet's resources, we need to inspire and engage the next generation. And what better way to do this than in finding amazing new species? I think this is especially significant in a megadiverse country like India, which has been noted as home to nearly 8% of global biodiversity. Discovery can also inspire governments and industry because it is positive. And there's just plenty of bad news environmental stories. The IPBES report, the UN report showing the amount of species that are in danger of extinction in the coming decades. Species discovery is inspirational. And the second reason I'd offer a focus on species discovery is because of this enabling role of technology, which all of the panelists have touched on. So we're really living in an age when discovery is more accessible than ever due to the declining cost of technology as well as its increasing availability. So many of the technologies that have been taken to the field have been discussed already. And I would add things like tracking tools like iNaturalist and eBird. Of course, environmental DNA has been touched on, artificial intelligence, drones, even social media coming into the field. These are increasingly cheap, sometimes free, they're portable. And there are platforms that enable and make available data sharing across multiple user groups, making large aggregated data sets freely available to more users. And here we have things like GBIF and the Map of Life. Further, technology has enabled and helped us determine where to explore and focus our limited resources and efforts. So qualitative models are helping predict where field expeditions should invest resources by estimating future taxonomic and geographic discovery opportunities. Discovery probabilities can be modeled based on a set of biological, environmental, and sociological attributes for different taxonomic groups, such as where discovery opportunities are best, for example, terrestrial vertebrates. No surprise, the Western Ghats comes up as a place for expected species discovery. So I'll close by saying that I think we need taxonomic initiatives to drive discovery and to fill gaps that have been mentioned here today, such as land invertebrates, plants, especially fungi. We need to engage non-scientists to care about the future of the planet. And I think species discovery is an excellent positive hook to do that. So I would suggest that we direct efforts towards enabling citizen science to join inventories, surveys, and research. And I fully agree with Dr. Uma that grants to include citizen scientists in inventories and field expeditions is a great way to go. And I would suggest that we encourage students and other citizen scientists who engage with these projects to build the attitudes, the skills, and the knowledge necessary to become stewards of the planet and contribute to solving real-world issues. And I will leave it there and look forward to the question and answer period.
Thank you very much, Catherine. Let me pick up on the last point. And a number of questions have revolved around open access to digital specimens and other data with Botanical Survey of India and Zoological Survey of India. I think there are at least 5 or 7 out of the 20 questions deal with this particular issues. So Dr. Mao and Dr. Kailash Chandra, if you could just take maybe a few seconds so we can get to other questions to address this very, very important question that is coming from the audience. I think people want to participate. People want to be a part of this discovery and exploration. And how can we enhance that? And so they're asking the question, when would this data be available publicly, the digitized specimens and other types of biodiversity data too? So Dr. Mao first.
Well, regarding the digitization of harbouring specimen, we have started working on it, and we have already digitized over 171,000 specimen. That is 171,000 like. So those will become put it up in the website soon, because our website, we have been working on it and it is almost ready. So because the earlier one, it crashed and it stopped working. So we are preparing a new one, and that is at the final about 90%, 95% is already completed. So just we need to wait for a few months more to get public. But if they write letter to us for any specimen, we are sending the image immediately to those who are interested. So we are receiving so many requests from different parts of the world or from Indian university and students. So whoever send the request, we send the image directly to them. That is not a problem. But for the public on in that digital herbarium, we have to wait. You have to wait for some more months. We are working on it, and once the website is ready, that will be whatever image we have, it will be all available.
Thank you, Dr. Kailash Chandra.
Yes, thank you, sir. With regard to this having access to anyone for our national zoological collections, as we all know, ZSI and BSI, both of them are this national repository of fauna and flora. As far as ZSI is concerned, first out of this 102,000 species which are known from our country, we have 17,000 type specimens. Already 12,000 specimens are digitized, and those photographs of all these 12,000 specimens are available in ZSI website. Anyone can have the access of the metadata as well as the pictures, which are very, very actually having a lot of our maximum our pixels. So they can enlarge it, and by any mode they can use. At the same time, we are just going to have the species page for 102,000 species that will have the name of the species, common name, entire diagnostic character, geographical distribution, then DNA barcode, plus the photographs of the different biotypes. So this, we are in process. We have already given this our contact to one of the agency in Bangalore, that informatics, they have been doing it for last 2 years, and around 30,000 they have already made it. So once at least We have, I think, considerable number of the species with us. We'll upload it to the GDSI. Even whoever is interested to have the research, they may write to us, and it's always accessible to them. We have been giving this our images and access to our specimen whoever is visiting to GDSI. Thank you.
Thank you. And there is a question for you, Uma. That's my decision to field questions to different panels, not necessarily for you. But Abhijit Jagdap, that in reptiles, very often you're dealing with species complexes. And as you know, there is also a related problem of cryptic species.
Mm-hmm.
So the question is, how can we address these issues?
Yeah, so that is a difficult issue, and that's one of the problems of trying to put organisms in bins called species, because speciation itself is a pretty continuous process. And so maybe in some cases, we will have to talk about species complexes and about lineages, leave it at that. That would be my suggestion.
Okay, thank you. So Ravi, are there a couple of questions you have prioritized? I shouldn't say prioritized, but which are common. Something for Catherine?
There is something on type specimens, but that's not for Catherine. Let me see. Maybe Catherine, you could take this. Is there any organization or body in India or globally which collates, explores, or serves as a compendium for the database of species diversity and richness?
Yes, thank you for the question. I can't speak to India, and I would defer to my panelists for that. There are a couple of international facilities. So GBIF is one, is an international network, a research infrastructure that's funded by the world's governments, and it is aimed at providing everyone, anywhere, open access data to all different types of life on Earth. And of course, things like GBIF or the map of life are only as good as the information that is fed to them. But those are two sources that are available to anyone, anywhere. And maybe there's some other specifics from the panelists for India.
Yeah, I think GBIF is the main organization where one can go to explore such data. I think, Ravi, I need to wrap it up and then give you a minute or so to conclude.
Maybe we can take just, this is something on capacity building. Are there training programs offered to undergraduate students related to advanced survey tools?
Let me take that question in terms of, as you know, in the mission, we have a separate component on capacity building, on strengthening the capacity of many, many institutions overall in biodiversity sciences. So there will be, once the mission gets implemented, a lot of opportunities, and I think a number of questions have arisen about citizen science too. So the response is the same. As you know, as many of you know, we have had a couple of conferences on citizen science. More activities are planned to get more feedback. And so there are going to be a lot of opportunities. I have been overall very much encouraged by this constant question coming up in all the webinars that how young people beginning their careers, or people who are thinking of the biodiversity science as a career, can participate. And I think there are going to be a lot of opportunities, and I hope some of the very, very inspiring presentations that were made during the last hour and a half would generate more interest. And so Ravi, I'm going to think, make some concluding points. I'm a stickler in terms of time. We are 5 minutes over time. So I just want to make some key points that have come out. And one is, which I'm very pleased to hear, that there are about 150,000 species in India that have been named. I think there seems to be a consensus. I've been struggling with that figure, and it's really very good to have that figure, that about 100,000 animals and 50,000 plants. Of course, we know that diversity is much more than just merely the number of plants and animals. There are major gaps. I think there are major gaps, not only in terms of discovery of new species, and discovery of new species can be very exciting, as Catherine mentioned, but also in terms of our understanding of ecological and evolutionary processes, ecological interaction and evolutionary processes. So biodiversity assessments are not merely going to an area and making a list of species. And even if you're going to an area and making a list of species, I think we have to think of new ways to assess biodiversity. How about people going together, plant people, animal people, looking at the biodiversity of selected ecosystems so that we have a better understanding of interactions. Right now, what is happening is we have data coming from plants, data coming from insects, birds, and then we have to look at, what are the patterns of distribution and abundance, and we have no ideas how they are interacting because the data are coming from different places. So this is the sort of work, which can excite people, which can excite citizens and young people, and help us in the program of discovery and exploration. A number of comments have been made. We also received a number of comments online about the open access, about data, how we can enrich the data, how we can build very comprehensive databases. People can participate in compiling those databases. People can also participate in contributing their expertise. All the data which we have is not necessarily accurate, and there are deficiencies in that respect. So wider participation, and Catherine mentioned that. And the utilization of new tools in terms of not only the DNA technologies and genomics, but in terms of predictive modeling, in terms of using artificial intelligence, where we should sample, because we have to do this work. We have to meet this Linnaean shortfall over the next 10 or 15 years. And I think we have to look very, very carefully for technology. National Geographic Society has a program on artificial intelligence and species discovery. If any of you are interested in looking at that, go to the website and look for call for proposals. But there are other possibilities too. So I think with those few comments, there are many more to be made. I think there are some people who have asked the question, what are we talking about? Biodiversity assessments. Our environment is withering away in front of us. Species are discovering. The forests are being destroyed. And I think these questions keep coming up during all the webinars. I want to assure people it has not escaped our attention. And I think the mission will address these problems. These problems are being addressed in other fora too. We have to remain optimistic, and we will hopefully turn the corner and make some progress. So with that, Ravi, It's all yours. And I want to thank all the panelists. Really, we are very, very grateful to you for your time. And to you, Ravi.
Thanks everyone. Just to conclude, a few announcements. The next webinar in the series is scheduled for Friday, 9th October, same time, 1600 hours Indian Standard Time. It's NISARG Bharat, Enhancing People's Participation in the ePBR Framework. And the one following that is on Monday, the 12th of October. It's Biodiversity in Agriculture, Will the Twain Ever Meet? We want to thank all of you for attending this webinar. For further information, please write to us via email, contact@biodiversitycollaborative.org. And as already mentioned, the remaining questions and for more detailed answers, we will get in touch with you by email. I would like to once again acknowledge the support we've received from the Office of the Principal Scientific Advisor to the Government of India for conducting these webinars. And I sign off by looking forward to meeting all of you next Friday at 1600 hours IST for the webinar on Framework for Electronic People's Biodiversity Registers. Have a good evening. Bye.
Bye-bye, Dr. Ravi. Dr.
Thank you
Kamal.
so much.
Thank you.
Bye.
Uma. Thank you.
Thank
Dr.
you.
Catherine.
Thanks.
Bye.
Bye.
Bye-bye.
Bye-bye.
Good night. Bye-bye. Thank you.